My name's Bronnie Tilbrook, and it's a pleasure for me to introduce the two plenary speakers this morning on this final day of the symposium. The first speaker is Sarah Cooley, who's the Director of the Ocean Acidification Program at the Ocean Conservancy in Washington, DC. Sarah started out as a carbon cycle researcher, and she early on recognised the potential for ocean acidification to impact marine life, and she's since authored many key publications on the subject, and through a role in the Ocean Conservancy, she's been a leader in informing policymakers and communities on the issues and actions that might address future impacts. This includes being a lead author in Working Group 2 of the 6th Intergovernmental Panel on Climate Change assessment report and many other publications. Sarah's a real star in the research community. She's recognised for her leadership and advocacy, communication, and her efforts and to bridge the sometimes very wide gaps between research and policy and the community understanding of ocean acidification and the potential pathways to address it. So Sarah will give a live online presentation. So are we ready?
Thank you to the hosts and the organizers of the Oceans in a High CO2 World meeting for inviting me to speak today. I get asked all the time how I can sound so optimistic when talking about global change and climate policy. The honest but complicated answer is that my optimism varies by the day. Some days I'm just angry and sad, and on others I have hope. But thinking about how ocean acidification knowledge has unfolded is something that gives me hope and ideas to drive other changes. That's what I'll tell you about today. On bad days when I feel pretty hopeless, I notice how climate disasters fill the top news headlines. I see acres and acres of land burned by wildfires, harming wildlife and people alike. I think about how weather patterns are changing around us towards greater and greater extremes of heat, cold, drought, and flood. I worry about freshwater supplies drying up and leaving communities parched and desperate. I worry about food supplies. How's climate going to break links in our supply chains? And who's going to be short of food then? Selfishly, I think about whether my own family is safe from the climate disasters most likely to hit our community. I've started talking to people in my community about the main climate-related risks we face together. In our area, it's increased rainfall-driven flood and exposure to heat-related illnesses. We also need to secure our homes and businesses against specific types of severe storms. I also read a lot of science fiction. I never used to. When I was growing up, a lot of science fiction focused on space exploration and situations that I couldn't relate to or I wasn't interested in. But today's science fiction often takes place in the future after a pandemic or a climate-related disaster. It's kind of strange that when I'm trying to find hope, I want to read more about futures that nobody really wants to experience. But it helps me picture what humans are up against, and it helps me think about what obstacles our societies will face. And over the last few years, I've spent a whole lot of time thinking about the scientific evidence about global change in the ocean. I co-led the writing of the Oceans and Coastal Systems and Their Services chapter in the IPCC Working Group 2 report released last spring. Our chapter team assessed thousands of papers to pinpoint the state of knowledge about climate impacts to marine systems, and we also looked at marine-focused adaptation. Not only was that job intellectually difficult, there was lots of information and lots of synthesis, but it was emotionally hard too. We had to sift through mountains of new evidence, even though we focused only on information published since the last IPCC report in 2014, and we had to count up just how much damage humans have caused to marine systems. We all struggled with climate grief, but that work also required us to look at the solution set. What actions people have proposed to help ecosystems and human communities withstand the climate impacts that are most likely. This helped us feel a little more hopeful. As you all probably know, my work has focused mainly on ocean acidification for over a decade. So naturally, when I think about ocean climate impacts and action, a lot of ocean acidification related examples come to my mind. And on this topic, we have learned so much in less than 2 decades. We've brought new communities into the topic, and we've created global momentum for research, observing, and policy action. There are definite lessons here that apply to ocean-focused climate action, and this means we have a deep well of experience to draw from. And we have an increasingly diverse global community that's paying attention to this and other global changes. The fact is, we aren't starting from a blank slate, and people are ready to see how invisible Earth system changes can be addressed. This is the foundation of my own hopefulness about working on climate change. There are lessons we can draw from the ocean acidification community and how the topic has grown that we can apply. And this is where the hope is. Detection of ocean acidification and attributing it to anthropogenic carbon dioxide emissions happened as an outgrowth of global ocean carbon cycle science. As repeat hydrographic surveys trace the movement of anthropogenic radiocarbon and carbon dioxide from fossil fuel burning through the deep ocean, models that incorporated this observational data identified that the ocean carbonate system was changing. Geologically speaking, this was a big deal. Human activity was changing the planetary buffer system that helps connect the fast and slow carbon cycles. Before this, a lot of people felt that the ocean was doing us a favor by taking up heat and carbon dioxide, and so it prevented faster atmospheric heating in those 2 ways. And people also thought dilution is the solution to the added carbon dioxide in the ocean, and that would be that. But the global ocean carbon observing community and the carbon cycle modeling community came together quickly and strongly to extend our knowledge. The first Oceans in a High CO2 World meeting in Paris in 2004 focused broadly on how ocean physics, biogeochemistry, geology, and ecology would respond to a high CO2 world. It paved the way for the multidisciplinary ocean acidification science we have today. Incidentally, as I looked it up, I found that the list of presenters in 2004 includes not one woman. I'm really glad that part of the Oceans in a High CO2 World legacy didn't last. Since about 2000, the ocean carbon cycle science community has built out our knowledge about natural and perturbed conditions in the ocean, and also looking at what the time of emergence is of global change and how gradual global change interacts with extreme events, which are also sometimes climate-driven. The scientific work of detecting and attributing ocean acidification gave rise to collaborations like the Global Ocean Acidification Observing Network, or GOON. GOON has more than 900 scientists from 105 countries, including people from government, public, private, and civil society organizations, all dedicated to improving understanding and exchanging information. These networks also interconnect local and regional efforts on acidification with work on other topics because of the broad affiliations and collaborations that members have. The lesson learned here is that this global scientific energy accelerated the creation of community and collaboration and teamwork. Compare, for example, if acidification was first recognized by a large private industry. Proprietary information might be critical for the success of that industry, and the spirit of inquiry around acidification would have been entirely different. This means that scientific energy can really help build community and collaboration around other global change topics too. There are also lessons we can learn from how knowledge of acidification's impacts on biology and how that has unfolded. Early on, acidification research identified that changes in ocean carbonate chemistry could directly affect calcified marine animals. Vicky Fabry's work in the Southern Ocean, pictured in the top, identified degradation of pteropod shells brought in to the lab for study. Coral reef experts like Joni Kleypas, pictured here at the bottom, identified how coral reefs, who were already suffering from bleaching that was driven by ocean warming, were also very likely susceptible to acidification damage. This still photo comes from a 3-minute video put together by the Heinz Awards, where Joni talks about the coral reef community's realization that acidification was an issue of concern. I highly recommend it. And not much later after this, oyster growers in the United States Pacific Northwest region experienced massive mortality of shellfish larvae that turned out to be associated with ocean acidification that was locally intensified due to episodic circulation changes. This kicked off a massive effort to identify how ocean acidification affected each marine species. By now, we have detailed knowledge for whole regions, such as Alaska. There are lessons learned from this part of the ocean acidification work that we can apply elsewhere. In the video that I screenshotted, Joni quotes Jacques Cousteau talking about how people will save what they love. Studies of ocean acidification's impact on marine life focused first on the systems that we love and depend on. The ocean acidification marine science community has emerged as a new block of fierce advocates for climate action who can point to specific impacts on treasured spaces and species. Ocean systems don't generally suffer from obvious disasters like wildfires or droughts, but things like ocean acidification are more invisible threats. And acidification is now a very well-defined invisible threat that we've been able to follow through from driver to impact. It was obvious from the start that ocean acidification didn't exist in a vacuum. It wasn't the only stressor in the ocean. Also challenging is the fact that ocean acidification isn't exactly like oxygen loss or temperature increase. When temperature or oxygen levels move outside of certain windows, conditions are immediately lethal. For some species at some life stages, ocean acidification can behave this way, but for others it doesn't. So to do good science, though, acidification had to be examined first as a single variable, which was the focus of many years of research, and that established our baseline about what we know. You might remember seeing this schematic. This one's from Sam DuPont, where at the beginning of ocean acidification studies, we congratulated ourselves on knowing a lot in a short time. We got discouraged when we recognized that it's more complicated than that. And our confidence in our knowledge has grown again as we recognize the real true complexities of this field. Ocean acidification doesn't have a clear-cut effect on one process or one physiological feature, and that's it. So the community continues to study the physiological processes that ocean acidification affects. And we find there's evidence that ocean acidification can affect metabolism, calcification, immunity, sensory processes, and neurochemistry. That's a lot. The assessment of our recent IPCC chapter is essentially that ocean acidification acts as a chronic influence on many biological processes, and it combines with different stressors. It can combine interactively, synergistically, antagonistically, either adding on, compensating for, or multiplying stress responses. The community is continuing to learn a lot about how responses of individual physiological processes, organisms, populations, or even species vary, and how these can result in ecosystem scale outcomes. This figure shows how two drivers can interact to create a whole different landscape of responses in a single physiological process. The lesson learned here is that even though global change impacts research is much more complicated than anybody would probably want it to be, we're getting a handle on how drivers result in responses. We're using a wide diversity of methods to study this, and this is giving us more confidence that our results are robust. But there are still lessons about humility and hope here. Both, we shouldn't assume too soon that we have things figured out when it comes to global change impacts, and also, we shouldn't doubt that natural systems will continue to surprise us again and again with their ability to cope and to adapt and to evolve. Humans are the ones that are often the most challenged to adapt to new conditions. Speaking of things we know about ocean acidification impacts and IPCC work, it's important to note that at this point we have enough information to begin to evaluate how specific ecosystems will respond. We also have enough information to begin to act. All of the individual and paired variable studies I mentioned have come together to give a greater picture of the nature of impacts on individual drivers and combined drivers on specific marine ecosystems. In the last few years, this knowledge we have on acidification as one of many drivers impacting an array of ecosystems has allowed us to begin to make projections about how different ecosystems will fare in the future. You might recall seeing graphics like this. We call them burning embers diagrams in the IPCC report. This is just an example of how we are increasingly being able to predict the climate-driven risk to particular marine ecosystems after we integrate and assess all of the information I've just talked about. And this is going to help us manage and use these systems sustainably. We can tell which ecosystems are more at risk in certain climate scenarios and where our attention should be focused in the short term and the long term. One of the first areas where people began to act on ocean acidification is in the shellfish aquaculture industry. This industry has developed just-in-time interventions like timing the intake of coastal water, adjusting water chemistry, or choosing when to place young shellfish out to grow. They're also working on longer-term interventions like breeding more resilient strains and developing markets for new cultured species. We're also seeing how communities with strong cultural ties to species at risk, like indigenous communities along the North American Pacific coast, are experimenting with approaches that draw from new and old techniques to increase food security and to help develop climate-resilient coastal ecosystems. Here's a photo of a clam garden in that region. A clam garden is a traditional structure that's used to culture shellfish that harnesses natural processes and increases the area that's favorable to grow shellfish. We're seeing local communities engaging students and resource managers in monitoring and involving local knowledge and indigenous knowledge to lead the work. For example, the Sitka tribe in Alaska is heavily involved in co-developing regional ocean acidification monitoring and responses for Alaska. The more experts that have been cultivated and have joined in the monitoring and the solution development and decision making, the more the solution set has become customized to specific places. And through this, we're seeing greater understanding about how acidification impacts whole ecosystems and how it is or is not implicated in ecosystem tipping points and other system changes. Last year, in Christoph Heinz's paper in PNAS, he discussed the quiet crossing of ocean tipping points, which basically noted that local systemic tipping points are likely to happen due to acidification, warming, deoxygenation, and other climate-driven processes, and globally, these local changes will add up to widespread changes with planetary implications. Over the years, my colleagues and I have written a good deal about what the toolbox of actions addressing acidification looks like. In 2016, my colleagues and I published a paper examining the way that OA action has proceeded, and we found that there were ways that all sorts of actors have engaged and pushed things forward. While doing the analysis then, 6 years ago, action tended to be the greatest at the individual and local levels, but since then, systemic changes have become more numerous. The lesson learned here that applies to other global change work is that even in the face of incomplete knowledge, we know enough to act. Acidification has shown us we need to recognize the many human connections to natural resources and local environments as we evaluate both the impacts and possible solutions. What helps one community might be completely inappropriate in another. And to identify the right way forward, we have to have everyone with an interest in the system at the table. This often stagnates because it's too hard, there isn't enough trust among participants, maybe we don't know who all should be involved, or different groups have diverging visions of what success looks like, or different risk tolerances. The approach and solution set of engaging many people and identifying the numerous human impacts, but also the options to take positive action that has been applied to acidification, it's a great study of inclusivity and multiple perspectives. Given what I said, it won't surprise you that locally specific actions that address acidification, such as treating water, examining new opportunities to co-culture macroalgae and shellfish, or relieving other stressors like nutrient pollution or pulses of freshwater runoff from the land. All of these opportunities have emerged and they're going to continue to be developed. And as we do this, we're bringing together expertise of thousands of people with local knowledge, indigenous knowledge, and new creativity in harmony with local culture and the natural system. This is absolutely fantastic, and it provides a wonderful model for other ocean-focused adaptation efforts. Success stories of implemented adaptations addressing all kinds of ocean climate impacts are starting to come out in great enough numbers that we can begin to evaluate trends. We assessed responses so far in our IPCC chapter, and we found that durable adaptation developed locally, coordinated well with other efforts to maintain healthy ecosystems and to develop societies equitably and sustainably, has a few common characteristics. It's always undertaken hand in hand with mitigation. It always seeks to solve many problems at once, not just one at a time. Nature-based solutions, for example, often fit these characteristics. And successful adaptation efforts rely on a variety of information. They're also aligned with cultural values, and they use multiple low-risk options wherever possible. And finally, they're inclusively governed. And the acidification community has worked hard to achieve coordination and collaboration that's inclusive and equitable. The International Alliance to Combat Ocean Acidification, or the Alliance, is a good example of coordination that is keeping adaptation and mitigation in view together. The Alliance brings together governments and organizations from across the globe dedicated to taking urgent action to protect coastal communities and livelihoods from the threat of ocean acidification and other climate ocean impacts. The Alliance explicitly cites mitigation as a goal. From their website, the Alliance says that members are working together to elevate urgency and ambition for climate action. They're integrating ocean actions across climate commitments and frameworks, and they're translating knowledge into policy actions and investments. Notice how the Alliance doesn't limit itself solely to ocean acidification. In fact, acidification is just the starting point of the Alliance's focus. And so in addition to providing these clear lessons on how ocean acidification and mitigation need to be attended to hand in hand, acidification policy work has provided additional attention on mitigation of CO2 emissions and strengthening that mitigation ambition from local to international scales. As I mentioned, the Alliance is bringing together ocean-focused groups to push for mitigation. And acidification provides this global to local to global spectrum, and with adaptation and mitigation tied hand in hand to use the examples for ocean climate change together. By now, I've made the case that the ocean acidification topic is a model for other global change issues. The work emerged from Earth system observations, but it got quickly paired with human dimensions as acidification's way of impacting ocean systems became clear. Collaboration was required. How acidification interacts with other drivers for individuals and populations and ecosystems has always been an area of focus, and it allows us to continue strengthening our understanding of possible impacts and consequences for not acting. People are coming together to save what they love and what communities depend on. As we have built out our understanding of how acidification impacts marine life, we're also understanding the limits of ecological adaptation and also humility on how complex the Earth system is. But we know enough to act, and we are acting. We are undertaking adaptation of human systems in collaborative ways that focus on justice and inclusion, and not in equitable ways that force a particular group to pay a price or be left out. And it's worth mentioning that we aren't moving at a snail's pace either. Some people have worried that focusing on global change science and research will trap us in some kind of research-only mode. However, we're already seeing ocean acidification begin to be considered in management guidance, again, for example, in Alaska in the United States. We know enough to act on acidification, even though the science is not 100% settled on every element. Being able to adapt and mitigate in spite of some uncertainty is a lesson we can share with other ocean climate and other realms. The growth of the ocean acidification community has shown that everyone has something to contribute to climate action. This global scientific energy has accelerated the creation of community and collaboration. There's been a strong focus on engaging people and building capacity through education and outreach efforts that span nations and cultures. We know from IPCC that adaptation has to be coordinated, it has to offer many simultaneous wins, and it has to be inclusive to be durable. We can apply our model of engaging people to other areas of ocean climate action, especially those that involve adaptation. And we're starting to see that when it comes to weaving ocean acidification into other international policy objectives. It all fits together. Thank you so much for your attention at this early hour. And once again, thanks to the organizing committee for this excellent conference. Although I'm not with you in person, I've been eagerly participating in this meeting all week. Feel free to reach out to me via email, which is listed below, or on Twitter. I'd be really happy to chat with you. Thank you so much.
All right, there's plenty of time for questions, and if you do ask a question of Sarah, would you please identify yourself and your institution? So let's start the questions. Are there any questions? Sarah, I've got a question. It's becoming quite diverse, all the approaches to address ocean acidification. So do you think is it bringing together the research community, or is it making it just so much more difficult to understand what to do in the future? What's your opinion?
You know, I think the oh, that's interesting. I've got an echo. If maybe the AV folks can help me out with that. I think the diversification of the research is it's tough to follow. That's for sure. It's hard to wrap your arms around all of it. And I think we're well past the days where one person can feel like they have mastery of the entire canon of work. But I think at the same time, as I was saying in my comments, it's showing us how we can be including other ocean changes into policymaking, into resource management, into research enterprise as well. We know so much about kind of keeping our eye on the ball when it's this big, complex situation. And making sure that information gets distilled and passed along to the right audiences who can use it. I actually feel like we have to set aside some of our expectations that we personally can track it all. And yet I think that the amplification and diversification is on the whole a good thing, and helping give the right people the right information when they need it. But it does come with a mental shift that we have to go through.
Yeah, thanks. Okay, John Pierre.
I'd be curious to hear from some folks in the audience about what gives them hope.
So if anyone wants to make a comment on that, please put your hand up. But the next, we have a question from the audience.
Great.
Hello, Sarah, Jean-Pierre Gattuso speaking. Thank you very much for this brilliant talk, and I like very much your energy and optimism. My question is a little bit peripheral to your talk, but it has to do with the IPCC. You have been also involved in IPCC reports. The IPCC is very prominent at COP meetings. Their sessions are very well attended by the delegates. Yet action is moving so slowly that it is really dispiriting. So, two questions. The first one, what is the future of the IPCC work, considering that so far it has been extremely useful to raise the issues and discuss solutions, but relatively poorly efficient? And also, what do you think of some scientists now in Europe are trying to get organized through an organization that is called Scientist Rebellion? What is your view on scientists turning into activism?
Boy, you're not letting me get off easy, Jean-Pierre. I think to answer the first question, I think that one of the challenges that IPCC faces going forward is that we have such detailed information about some areas of the globe. And in those areas, we already know enough to act. In other areas of the globe, we don't have as exhaustive information. So, for example, one of my IPCC writing colleagues, Aditi Mukherjee, who was a CLA for the water chapter, was pointing out that in her part of the world, in India especially, they don't have the level of local information about hydrology and water supplies that is needed to make decisions. So there are places in the world where we still need a good deal more information to support evidence-based decision making. So I'm hoping that in the future, IPCC work will sort of even things out a little bit, and there'll be a greater emphasis on the areas and topics that we don't know as much about. So, for example, in this assessment report cycle, we saw a whole lot more involvement and information from social sciences. And that has really opened up a new chapter in our discussions about what solutions are appropriate for people and what are the impacts on human well-being. So I'm looking forward to seeing more of that in future IPCC work. But I do think that there needs to be sort of a conscious rebalancing of, like, let's not keep delving into, getting from, very high confidence to it is certain, in some topics, because there are other topics we really need to know a lot more about. And I think regarding your point on decision making, I am hopeful that the additional information we have on human well-being that's coming out with additional assessments, I'm hopeful that will make the information concrete enough and kind of personalized enough to really help continue driving additional urgency in the decision-making and policy space. Because when you put things into human terms, the decision-making calculus really changes. And then your question about scientist rebellion, I've been watching that with interest, and I've been thinking about, is that a role for me? Is that something that I should be involved in? And personally, I made a different decision. I decided not to get involved with that because part of my job description is to be sort of a spokesperson for the ocean and a spokesperson for my organization. And personally, if I'm, getting arrested, chaining myself to the front door of an international bank, that's not going to drive forward, my job description or the things that I sort of see for myself. Personally, I prefer to make a difference through, discussion and negotiation and personal contacts. That's just my style. I mean, those of you who know me could agree, I think. I'm not going to skydive with a big banner behind me, but I think there's a place for everyone, and folks who are moved to be involved that way, that can make a difference. I think we all need to bring our personal selves into this fight. It's no longer sufficient to say, well, I need to be objective. No one is objective, especially when we're talking about an existential threat to the way that human beings live and thrive today. So I guess those are sort of my thinking. It's hard. I can't read the room, so I don't know how you guys are receiving what I'm sending here.
All right, are there any more questions for Sarah? All right, Sarah, thank you very much for your great plenary, and maybe we could all thank her again.
Thank you, Bronte, for that nice moderation.
Thank you. So we're ready now for the second plenary. The next plenary will be delivered by Guillermo Diaz-Pulido. Guillermo, he completed a science degree in Colombia and before moving to Australia to attain a PhD in marine botany at James Cook University in Townsville. And he's, for a number of years, he's led the Coral Reef Algal Laboratory at Griffith University in Brisbane. And among other work, he's also a lead contributor to the effort to restore degraded reefs on Australia's Great Barrier Reef. He's a highly regarded authority on the role of macroalgae in coral reef ecosystems, and he's published extensively on the subject. This includes research on the impacts of ocean acidification and warming on the calcification and growth of crustose coralline algae, which is so important in the structure and functioning of coral reefs. So, Guillermo, over to you.
Thank you, Bronte, for your kind words. Buenos días a todos. Good morning, everyone. So it's a great pleasure to be here. Just wanted to, first of all, thank the organizers of the symposium for inviting me to give this plenary talk here. So what I wanted to talk about today is to discuss the effects of ocean acidification on benthic macroalgae in coral reefs, and then try to understand what are the consequences of these changes in abundance of macroalgae for the ecology of coral reefs. So I have organized my talk to discuss first the effects of ocean acidification on crustose coralline algae, or CCA. I'll be using the term CCA often in my talk, so you better get familiarized with that. CCAs, crustose coralline algae, Bronte just referred to them. And these are these pink crusts growing here amongst the corals. And I'll be talking more about them in a minute. And then I'll focus on the effects of ocean acidification on algal turfs and fleshy macroalgae. But before I get into the matter, I just wanted to give you a little bit of background about the importance of coral reefs. I know all of you are familiar with them, but I just felt the need to emphasize their importance, just to making the points that these are ecosystems of extreme beauty, right? Extremely high diverse, very productive. They're home for millions of species. Many of them are economically important. They also provide livelihoods for millions of people living in coastal communities. For example, in the Great Barrier Reef, the Australian economy takes around $6.1 billion from the reef. So they really, really depend on coral reef health. Now, in terms of the importance of macroalgae in reefs, there are the primary producers. They contribute to primary production. They fix carbon through photosynthesis. They produce oxygen. We see here this green alga, Colerpa, producing lots of bubbles here, contributing to biogeochemical cycles. The carbon fixated by these algae are then flow through the food chain. Food for grazers, this beautiful photo from NOAA here where we see all the grazers in the reef, herbivorous fish, parrotfish, surgeonfish, feeding on the biomass produced by the microalgae. But I think one of the most important roles of microalgae in the reef is their contribution to reef construction, reef cementation. And that's the role played by CCAs, or cross-coralline algae. You can think about, and we see lots of them here in this shallow, gorgeous reef in the Great Barrier Reef. You can think about their contribution like a building. So in a building, we have the blocks, right, the bricks, and the corallines, or the CCAs, are kind of the mortar or the cement that is really holding those bricks together. So that's exactly the contribution the coralline algae make to the reef. Now there is another thing, and is that the CCAs induce the settlement of coral larvae to the point where many coral larvae just exclusively settle on specific coralline algae. So therefore they contribute to reef resilience, reef growth, reef population, coral population recovery. Macroalgae, fleshy macroalgae in particular, also are very important for buffering the effects of ocean acidification, and Jean-Pierre has talked a lot about that. So that's one of the contributions as well. They take up CO2, they modify the chemistry of the water. So that's another important sort of role macroalgae play in coral reefs. Macroalgae have been, are also indicators of coral reef health. And we know this because many coral reef ecosystems that are healthy, like this one here, where there are lots of corals, lots of coralline algae, due to a variety of different processes, they shift into this state dominated by macroalgae, as we see there. And there are many different processes that have been involved in these transitions, these shifts in dominance from corals to macroalgae. You could think about reduced herbivory, for example, due to overfishing or diseases of urchins and so on, eutrophication, nutrient enrichment, the algae just grow out of control, taking up all those nutrients, overgrow, kill the corals. Coral mortality, like for example, due to bleaching, corals die, then get colonized by macroalgae. And now, ocean acidification has also been implicated in this process here due to a CO2 enrichment effect on macroalgae. The other consideration that we need to keep in mind here to understand the effects of OA on macroalgae is that macroalgae are extremely diverse group of organisms. There are literally thousands and thousands of species of macroalgae in reefs. So because that complexity, I feel the need to group them into functional form groups so we kind of try to synthesize their effects on these groups. So we've got the CCAs again here, couple of deep water species here, and community of shallow water CCAs. We've got the algal turfs, which is a community composed of minute, tiny little filamentous algae, very productive, very important for the production of food for grazers. And we've got the upright macroalgae, both the calcareous, the calcifying algae like Halimira, this is a very important alga for reefs. And we've got the upright fleshy macroalgae like Sargassum. I'm sure many of you are very familiar with this fleshy macroalga here. So let's get into the matter here, talking about the effects of OA on crustose coralline algae here. But before I go into detail, again, I just wanted to mention that different macroalgae have different types of calcium carbonate. So we've got the coccolithophores, although they're of course not benthic macroalgae, they precipitate calcite, right? And the solubility of that calcite is very low, and we've heard a number of talks here discussing the effects of OA on coccolithophores. We've got Halimira. Halimira precipitates aragonite. Aragonite is a polymorph which its solubility is intermediate. We've got the coralline algae, CCAs, that precipitate high magnesium calcite. And one of the important things of the calcification of CCAs is that their calcification happens in the cell wall, which is different to calcification in the other organisms, which for the coccolithophores was intracellularly. For aragonite happens in between the cells, while here, the corallines, they do precipitate in the cell wall. So that's a different mode of calcification. Now, corallines have also another extra feature here, and is that they precipitate other types of minerals like dolomite and magnesite, which have a very different solubility here. They're extremely, extremely robust to changes in carbonate chemistry. And because omega is a function of the solubility constant, which varies with the carbonate type, then omega is different for each of those types of minerals. So that's something that we need to keep in mind. Again, here, just giving you a little bit more of context and background so we can understand the effects of OA on calcification on the corallines. This is Porolithon oncotis. This is our lab rat. It is, I would say, the most important alga in the reef. It really helps cementing the reefs in the shallow reef environments. This is so like a cross-section of the alga. We're talking about here 5 millimeters to 1 centimeter thick. And we see that here is the pigmented, pigmented tissue where photosynthesis is, and then underneath that's half a millimeter of, around 100 microns of tissue. We have the white skeleton, which is still a living alga, but it doesn't have the pigments. And we've done some examinations of the mineralogy of the algae here, and we see that in the upper layer, the upper pigmented tissue, the predominant type of calcium carbonate is high magnesium calcite. Underneath this upper layer of pigmented tissue, we get a lot of dolomite, which, as I mentioned before, is very robust to ocean acidification. And then we have pockets of other minerals here, like, for example, magnesite, few pockets here and there, and the corallines also have aragonite, sometimes inside the conceptacle chambers, the reproductive chambers. So as you see here, the mineralogy of these organisms is extremely diverse. Yep. And with all these different minerals having different solubilities to ocean acidification, so that's something that we need to keep in mind. Now, the other important consideration here is that the coralline algae are able to control the carbonate chemistry in the calcifying fluid. And this is some research that is just starting to emerge from the the work by Chris Conwell and colleagues, and also Riz et al. And we heard a talk yesterday, a really interesting one, trying to examine to what extent the algae can actually control or regulate the internal chemistry, right, in the calcium carbonate. So altogether, if we want to understand the effects of ocean acidification on coralline algae, we need to have several considerations. One is we need to consider what happens in the water column, right? Omega with respect to seawater, omega within the calcifying algae with what happens inside here, and also because I told you that they calcified in the cell wall, so we need to understand what goes on, or what are the constituents of the cell wall, which are primarily monosaccharides, polysaccharides, okay? So I just did a quick search in the Web of Science using the words coralline algae and ocean acidification. Nearly 400 papers came up with 24 reviews, 5 reviews in the last couple of years. So this is a field that is moving very, very rapidly. Lots of people have contributed really awesome research here to provide this framework that we need so we can understand the effects of ocean acidification. Now, when we try to understand what goes on with the coralline algae, we ask the question here, well, what sort of responses are we going to measure? What sort of metrics we need to consider to understand those effects? So we could think about population level responses like cover, recruitment. We could think about community composition, shifts in species, for example, or we get more into the physiology of the algae, looking at the effects of photosynthesis, respiration, or effects on mineralogy, calcification, or we get more into the effects on the health of the algae, quantities of pigments, or bleaching, for example, or we get more into the ecological interactions, whether there are shifts in dominance, for example, some species overgrowing others, and so on. But here in this talk, I just wanted to focus on the population level responses, just cover and recruitment and calcification, because I think these two metrics or responses are probably more directly related to the role of calling algae as cementers in the reef. And one of the first demonstrations of the effects of ocean acidification came from this brilliant work by Ilse Kavner, where she grew different artificial substrates in water with different pH conditions. And in the controlled treatments, she found here high cover of CCAs, lots of pink crusts here, high number of recruits, while in the acidified treatment, very low CCAs, lots of turf algae. At the same time, we're working with colleagues from Ken Anthony and Ove in Heron Island in the Great Barrier Reef, where we grew adult calling algae this time at different PCO2 or pH conditions. And what we saw was a very significant decline of calcification as we increased the acidity levels of the water, to the point where the callings actually were dissolving. Right? And this contrasts with the effects on the corals, because although the corals did calcify less, they did not dissolve under acidified conditions. So we concluded here that CCAs are very, very sensitive to ocean acidification, much more than the corals. This upload here from Chris Cornwall's review came up just very recently, where they examine all the effects of OA on CCAs, particularly focusing on CO2 SIPs. And we see here that as the water gets acidified closer to those CO2 SIPs, we see a reduction in the cover of CCAs, and that's building up on the great work from Jason Holt-Spencer there in the Mediterranean. And they also concluded here that recruitment of CCA is also very sensitive to ocean acidification. And you see here how the corals recruitment goes to very low levels at the reduced sites. Couple of photos here from Catarina Fabricio's work in Papua New Guinea. Couple of settlement tiles here. This one at the control sites. You see lots of coralline algae there, lots of pink crusts in the control sites, while in the CO2 SIPs, there is very few to no coralline algae here. So they hypothesized there that competition with the filamentous algae, it's driving these transitions. So we actually tested that hypothesis there, and we found that it is not so much the competition with the filamentous algae that is driving that change, but changes to the germination rate of the spores of the coralline algae. Very few spores actually germinated in the high CO2. Many of those spores were actually abnormal, and this is just compared to the ambient control. But nonetheless, there was very little recruitment, very few spores germinated in the high PCO2 treatment. Now, so despite those general trends that we saw in that meta-analysis from Chris Cornwall, we need to also consider the variability between species. And this is some work we are currently doing, where we are comparing calcification across a variety of different species. This is just an example where I picked up 3. Again, our lab rat here, Porolithenum cordis. We see this super sensitive alga Lithophyllum, which is just very sensitive to ocean acidification compared to the others. And this other alga here is Porolithon, is just very robust to ocean acidification, one of the most robust CCAs that we're currently working with. Now, and I just showed you here calcification rates. Well, here I'm just talking about growth. And we see that the responses are very different. Sporolithin, again, is very robust, not so, there are no significant changes here with sporolithin, and the way we measure growth is by we get the coralline algae, we stain it with different stains, there is a mark here, and then we could quantify how much of the alga is actually growing. And we see here that for sporolithin, there's not much change. Again, sort of supporting this idea that this alga is super, super robust. The other thing that I wanted to point out here is that we could actually get different outcomes depending on the metric that we are using, whether we're measuring skeletal dissolution, for example, or whether we're measuring growth or net or gross calcification and so on. So that's something that we need to consider. So this is a page in our group, one of our great students there, so she was interested in trying to understand why we see those differences in sensitivity of the algae to ocean acidification. So again, so she tested here porolithin and sporolithin, and yeah, confirmed that yes, they're very sensitive. Photosynthesis went down, a combination of low pH and high temperature, and oops. And also she did some transcriptomics work, where she found that around 400 different genes are either upregulated or downregulated in this alga. So it is a very reactive alga to changes in the acidity of the water. But the most interesting thing really here is that she found that sporolithin actually had no significant change in the upregulation or downregulation of the genes. And again, this is providing some really, really exciting some insights into the potential sort of mechanisms by which these algae could adapt or to changes in the environment. So this is really fascinating. This is just a heat map, looking here at the number of genes that were upregulated in the high CO2, high temperature treatment. Some genes were downregulated. She then went into trying to link those transcriptomic responses with the cellular processes. So, and there are many genes involved in the Calvin cycle and so on, but I don't want to get too specific here, but the paper has just been accepted and is a really, really interesting way of understanding those variability in the sensitivities of algae to ocean acidification. Now, we've been doing some work on the mineralogy of coralline algae and how does ocean acidification affects what goes on inside the coralline algae. Just reminded you that the coralline algae have different mineralogies, right? In the surface, the pigmented tissue compared to the bulk, to the unpigmented tissue. So when we exposed this algae to different PCO2 levels, we saw an increased accumulation of dolomite under the combination of high CO2 and high temperature treatments. So the algae, the mineralogy of the algae is really changing internally when we expose it to different CO2 conditions. Aragonite was actually much higher in the cold water treatments, the low PCO2, and high magnesium calcite, which is the dominant mineral of this alga. Didn't change much there in the unpigmented tissue, but they took up more magnesium, right, as with increased temperature. So the point really here is that the mineralogy could potentially be very responsive to the changes in ocean acidification. All right, sort of shifting gears here a bit, I would like to touch on the effects of OA on algal turfs. And I guess the story with the turfs is slightly different because they are very responsive to the mortality in the corals. So this is typical coral reef in the inshore reefs of the Great Barrier Reef. When they get hit by bleaching, many of those corals die from bleaching. Not all corals die from bleaching. And those that die get colonized by filamentous algae. So we see these transitions in many reefs around the globe. These are data from the Australian Institute of Marine Science, where we see the corals here in black. It just collapsed right after a mass bleaching event here. And then we see this phenomenal increase in turf algae. Also, this data here from Palmyra Atoll, from Jen Smith's group in California, we see similar increase in turf algae in response to changes in the corals, in the coralline algae. So turfs are very responsive to what happens to the corals. And we now know that the frequency of bleaching events per decade is going up. It's projected to continue to go up with CO2 emissions. This is the data from Goldberg's projections. And at that time, he projected that reefs would be experiencing, we are somewhere here, around 4 or 5 bleaching events per decade. And that's exactly what's happening now in the Great Barrier Reef. Just the Great Barrier Reef has experienced 6 bleaching events in the last 15, 20 years, some bleaching events back to back. So that's, these projections are really tracking very nicely what's happening. So we know bleaching is going up, frequency, intensity of bleaching, that's leading to more corals dying from bleaching. We know that turfs love growing on dead corals here, dead coral here. So does that mean that we'll see more dominance of turfs in the future? Most likely, but it really depends on the rates of the recovery of the corals, the health of the ecosystem as such, grazing rates, how good the herbivores are there to control the algae, but also on the responses of turfs to ocean acidification. And we've done some work on that space. This is work by Dorothea Bender, where she looked at species composition of this community growing on dead corals. And she exposed those turfs to different CO2 conditions. And what we saw there, what she saw there, is an incredible proliferation here of cyanobacteria under the high CO2 treatment. There were some shifts in species composition. Some algae didn't like those conditions or were potentially outcompeted by the cyanobacteria. So overall, what we see in the responses of turfs to OA is that there is shifts in species composition, increased growth rates of the turfs, increased biomass, and so on. This data here is from Ben Harvey. He was presenting a couple of days ago in one of the sessions here, where this is just plotting here the changes in macroalgae as reefs get acidified in CO2 seeps, decrease in macroalgae, increased in algal turfs. This is not so much of a coral reef here. The coral cover is only like 10%, but nonetheless, there's increased support here for a shift in community dominance from macroalgae and corals to a turf stage. And this is becoming kind of more frequent, particularly in kelp beds as well. And here, the mechanisms that are postulated here is that this turf algae, these filamentous turfing algae, trap a lot of sediments that causes a hypoxic condition in the turf community, and then sort of impedes germination, growth of coralline algae, and the corals. So it seems that turfs are one of the winners under ocean acidification conditions. All right, shifting gears again, just gonna move to discuss a little bit the effects of OA on fleshy macroalgae. And we see this very gorgeous community of benthic macroalgae, green seaweed, some reds in a shallow coral reef there. Again, I did a quick search in the Web of Science, crossing using the words macroalgae and OA, more than 400 hits there. 34 reviews of the literature, and these reviews are really useful for giving us, like a summary of what's going on in the field, identifying trends, synthesizing all the information. There's some really, really great work from a lot of people in this field. And one of the outcomes of those reviews really is they all emphasize that the effects are very variable, extremely variable. It depends on the species, whether they are fleshy macroalgae or the upright macroalgae. All the effects really depend on the species. This is some really nice work from Maggie Johnson in Jen Smith's lab, where she's here looking at the changes in growth, in weight for a variety of different species. We see that some species don't really like the acidified conditions or the growth conditions in general, but some species actually do very well, like some of the browns, dictyoras, do very well under acidified conditions. So there's a lot of variability in this group. Now, CO2 seeps have provided an interesting sort of analogous of what could happen in future ocean acidification conditions. And this is the work by Katarina Fabricius here in Papua New Guinea, in the northern part of Australia, where they discovered these CO2 seeps, and they looked at community, they looked at community variability, and they documented shifts from coral-dominated systems to algal-dominated systems very close to the CO2 seeps. There was a double in the amount of macroalgae, although the doubling there was only from 2% to nearly 4%, not much of a shift there, though. However, this is work from Ian Enoch, I think he's here somewhere in the audience. Really nice work here, documenting much more dramatic shifts in macroalgae. As sites get closer to the CO2 seeps, we see they saw transitions from coral-dominated systems to macroalgal-dominated systems, right? So there's some really, really interesting shifts there in dominance from corals to macroalgae in these sort of naturally acidified systems. So in trying to provide sort of like a physiological framework to understand the effects of ocean acidification on macroalgae, and the question here is, can the CO2 uptake physiology explain the responses of macroalgae to ocean acidification? And we need to revise a little bit a phycology class here. Macroalgae have a variety of different mechanisms by which they take up CO2 to the site of photosynthesis. So they use direct uptake of CO2 via diffusion gradients, and the expectation here is that those species that have this type of mechanism would do very well. Now, some macroalgae also take up bicarbonate, and they use a variety of different mechanisms to convert that bicarbonate to CO2 at the site of photosynthesis. And this is just an oversimplification of those mechanisms for the sake of this talk here, but the responses would be, for some species it would be variable, for some species would downregulate those CCMs, and using those carbon concentrated mechanisms is very costly. So it's a lot of energy goes into using that. So by having this excess of CO2, those macroalgae could actually down-regulate the use of the CCMs. So Cornwall here tested this hypothesis whether we could use this framework to understand the responses of ocean acidification. And in fact, they supported this hypothesis when examining the shifts in macroalgae in the CO2 seeps in the Mediterranean. However, other studies have challenged that notion here, and they argue that yes, although we can use this scheme to understand some of that variability, we still need to do much more work to understand the context of the different studies. For example, we need to understand levels of light in the environment, which could sometimes be much, much more important than the actual physiology of the algae. We need to understand the context of nutrients, and so on. So although this framework is really useful for understanding some of those effects, there is much more that we need to do to fully understand that variability. All right, so just shifting gears again, I just wanted to explore how those changes that happen to the macroalgae in reefs could impact the ecological interactions in reefs. And we've done some work looking at the impacts of ocean acidification on the competition between the macroalgae and the corals. And we've grown pairs of species in the lab where we put the corals in contact with macroalgae, because we know that macroalgae are very good competitors for space against the corals. And what we've seen here is that under high CO2 conditions, the macroalgae become very, very aggressive, right? So the corals really suffer here under the combination of high CO2 and having the macroalgae. And we've tested this a number of times with different species, red algae, green algae, brown algae, different CO2 levels, and we always see that that combination of macroalgae with high CO2 is not good for the corals. We see high coral mortality there. But we're wondering what sort of mechanisms are taking place there. How is that mortality of the coral happening? And we know that the macroalgae compete against the corals using a variety of different mechanisms. They could just grow a lot, right? Overgrow the corals, suffocate the corals. They could potentially abrade the corals. There's actually transfer of microbial microbes from the algae to the coral, and that triggers diseases in the corals. Also the DOC, the dissolved organic carbon release hypothesis, where the algae release a lot of DOC, and that fuels microbial growth on the coral, and that really alters the health of the corals. So we wanted to test some of these ideas in our lab, and here we hypothesized that ocean acidification would intensify or would increase the potency of some of those allelochemicals, and then we'll see high coral mortality there. And this is some really interesting work we did with Mark Hay, where we got the seaweeds, we extracted the chemicals from the algae, and then we inoculated those chemicals into some gel pads that we put into the corals to see what happened with those allelochemicals. And in fact, what we saw there was a reduction in the photosynthetic ability of the corals when they had the extracts of the macroalgae grown under high CO2 concentrations. So it really sort of demonstrated that the chemicals produced under high CO conditions became more potent. That's very interesting. However, this was not universal. The other species did not show that trend, which suggests that there are other mechanisms there that might also be taking place. So, for example, DOC. Remember that I just told you that the release of DOC could trigger microbial growth on the coral, which causes a decline in the health of the coral. So we wanted to explore whether DOC is increased, the release of DOC is increased under high CO2 conditions. So we've got here data for several species growing under 2 CO2 conditions, ambient and acidified conditions, and what we saw was a phenomenal increase of DOC when we grew the algae under the high CO2 concentration treatments. So our interpretation of this is that yes, the algae are using the CO2 for photosynthesis, for growth, but there is, they cannot handle all that CO2 that is being fixated via photosynthesis, so they fix it, and with the excess of that CO2, that's being released back into the environment. And that's why we see this phenomenal increase of DOC production that goes out into the water. And this would have very important implications for microbial biochemical responses, for microbial growth, and potentially for space competition of the macroalgae over the corals. All right, just gonna have a glass of water. So just in the final couple of slides of my talk today, so just wanted to briefly discuss some of those implications of the increase in macroalgal growth for reefs. So we've discussed already shifts in species composition, moving from coral-dominated systems to macroalgal-dominated systems, but also the calcifying algae can get replaced under those conditions. There might be shifts in reefs from net accretion, net growth in reefs, to net dissolution, and that's because the corals get affected. There is not enough calcification in the system. We have more seaweeds here, and so the habitat is being lost. Is lost in complexity. If there is loss of complexity, then fisheries could potentially be impacted. Corals don't like settling under these conditions here where there are lots of macroalgae. They find it really hard. So potentially there would be a decline in the settlement of the coral, which could be translated into reduced coral population growth, reduced in reef resilience, the ability of corals to recover, and so on. And I didn't really cover any influences on trophic dynamics here in the reef, but that's also to be expected when we see these shifts in community composition in reefs. But I believe that one of the most important implications for these shifts in community composition in reefs would be by losing the coralline algae here. Remember, as I mentioned at the beginning of the talk, reefs are really maintained by that cementation of the coralline algae. If there is not that cement or mortar in that building, in that framework, then corals would become the reefs would become very fragile. And that would be very, very sort of problematic for many communities in the Pacific which rely completely on the shallow coral reefs, which are dominated completely by coralline algae, like this species there, porolithen. So losing the corallines would have very significant implications for reef accretion, reef growth, which would impact coastal communities, and importantly would be a potential loss of protection against storms. But also, and I didn't really cover this in this talk, is that if we lose the corallines, that substrate, which is preferred by many coral species, would potentially impact on the ability of those reefs to recover. So we now know that some corals do prefer this species of coralline algae called Titanoderma. This species is very sensitive to ocean acidification from the work here of Chris Doropoulos. So losing this species, which is a preferred substrate for many coral larvae to settle, that could potentially have ramifications for the resilience of the reef. This is just my last slide, so I just wanted to sum up some of those effects of OA on coralline, on the algae, on benthic macroalgae. Reduced cover, reduced recruitment, reduced calcification. That's sort of consistent across the group, although there are some species that are quite robust. There are changes in the mineralogy of the algae. Overall, this is a very sensitive group to ocean acidification. Algal tephs would potentially continue to dominate many reefs through to shifts in species composition, proliferation of cyanobacteria on those algal teph communities. With fleshy macroalgae, just very hard to generalize given the complexity, given the diversity of species there, but most likely would be an intensification of the competition with the corals via a variety of different mechanisms. And I just a minute ago mentioned the potential ramifications for the ecology of the reef and losing reef resilience, which would impact, the economic, the services that people get from those reefs. So I've just reached the end of my talk. Thanks very much for listening.
Hi, Guillermo. Thanks a lot. I was just wondering, taking into consideration what you just showed us in terms of the sensitivity of the CCA, to ocean acidification. And you know, there are like globally a lot of efforts in terms of coral reef restoration. There are a lot of coral restoration programs, and they actually are investing a lot of money and efforts in this kind of like projects. So do you think they should actually be also taking into consideration like a parallel program in, I don't know, maybe try to find like more resilient strains of CCA to low pH and try to start also cultivate these strains in parallel to improve the success of these coral reef restoration projects? Or what are your thoughts in this sense? Thanks.
Thanks, Celeste, for that comment. Look, we are heavily involved in a project looking at reef restoration in the Great Barrier Reef, especially try to understand that interaction between the coral larvae and the coralline algae. And one of those species that we're focusing most of our efforts is on Titanoderma, that alga that I show you there, which is really, really good for inducing the settlement of those corals. And so we're trying to grow this algae. We're trying to identify growth conditions for coral aquaculture. Now, there are different limitations there, one of which is that we don't really know the systematics, the taxonomy of many of these species of coralline algae. So that's one of the major limitations. So if we want to, let's say, try to cultivate one strain that might be more robust to ocean acidification and warming, we first need to understand, who are we dealing with. And the coralline algae have just been, we're heavily working on that as well, trying to understand the diversity of coralline algae, and there is an incredible diversity of coralline algae. The alga that I talked to you about a lot, Porolithon onchoris, turn out to be at least 20, 30 different species of Porolithon onchoris. So if we want to work towards developing or culturing strains that are robust to ocean acidification, we really need to take a step back and provide even more fundamental science before we could then move forward and say, okay, well, is this a strain of coralline algae and that is more robust that we could just culture, for example, and potentially think about potentially repopulating reefs or things like that. But that's an active area of research, yeah.
Hello, Guillermo.
Martin.
Thank you so much for the presentation. I would like just to note your opinion about the coral reef located in the tropical Pacific in the oxymominous zone, okay, nearby. This guy, I don't know how they are living. They are sleeping with the enemy, right? Because just some 40 meters, the oxymominous zone is located in the Mexican side. But Colombia, where Celeste is coming from, I'm talking about Isla Gorgona, the Mexican coast, you can find the coral reef. In your expertise and the physiology, what is your guess about that?
Yeah, well, I don't know much about the physiology of the corals, but I could tell you a little bit about the physiology of the coralline algae, and from the work Marcelo has done on that area, we know that the coralline algae there are present, right? But those corallines are very thin, right? They cover a lot of areas. So for example, in the Galapagos and Gorgona, some of those places in that area. Reefs do have the corals, of course, but there's also a fair bit of coralline algae. But those corallines never form these sort of thick crusts that we see in more tropical sort of locations at the Great Barrier Reef or the Central Pacific. There, those coralline algae are extremely thin. Could potentially be different species, more robust potentially, those areas are of upwelling and so on. But yeah, so I think there could potentially be some adaptations there to cope with low levels of pH, potentially high levels of nutrients, but those communities haven't really been studied from the coralline algae perspective and their contributions to reef growth. So I would assume that, yeah, those CCAs would potentially have some interesting strategies to cope with those different conditions, even either through taking up more nutrients, for example, right, or modifying the chemistry in ways that we still don't really understand.
Thank you. Well, a great job. I enjoy a lot. I'm very interested about the mineralogy. I think it's so interesting. And my question is possible that one species of this calcifying can change under the stress, the low pH, the mineralogy, and go to a more powerful, like dolomite on that, in order to not to be the species that can in situ make the difference, the champion under this condition. And the other question is, what about this kind of algae in the calcifying algae in the upwelling system, like low pH, that can be very interesting for research, no? And to compare and about your research.
Yeah, look, for the first part of the question, well, we've seen that the mineralogy of the algae is very responsive, not for all the species, at least the species that we've worked with, Porolithodon oncotis, the mineralogy is very responsive. And we see increased accumulation of dolomite. We don't know the mechanisms by which that happens. And there are a lot of work on the dolomite sort of side of carbonates. It could be that the algae in itself is changing the polysaccharide composition, the constituents of the cell wall, to something that is more conducive to more precipitation of dolomite. That's one of the hypotheses. The other is that there is increased microbioerosion. There's a lot of microbioerosion under acidified conditions, and those microbes would be munching on the much softer high magnesium calcite, leaving behind the dolomite, which is much more robust. And therefore, we see an accumulation of that dolomite. And we get into a number of other more crazy hypotheses there. But yes, the mineralogy is very reactive, but not for all the species. But we still don't understand very well what's happening there. For the other part of the question, we in fact have a project in Colombia, in the Caribbean coast of Colombia, where there is an upwelling, a coastal upwelling there, and we have been following the coralline algae communities, the growth, the calcification in those upwelling impacted reefs. And what we've seen so far is that, contrary to what we expected, I mean, we hypothesized that, okay, well, the upwelling brings low pH, right? Therefore, we know that calcification is sensitive to low pH, so we were expecting lowering in the calcification rates of corallines during the upwelling. Well, in fact, we actually saw the opposite. And we're still, again, trying to understand what's going on. A couple of hypotheses there is that upwelling brings more nutrients. They are plants. They love nutrients, right? So they could use them up as sort of like an enrichment effect, or they could also be an enrichment effect of PCO2 on the algae. Again, algae need CO2 for photosynthesis. But we still don't understand very well whether that's an enhance of gross calcification, like the new growth of the algae, or whether it's actually dissolution, what's going on what's been influenced by pH. So that's a project, an ongoing project, and there are lots of things that we could learn from those communities in these upwelling reefs.
Thank you. Any more questions? Actually, I've got one. So, Guillermo, when you go through the barrier reef, particularly in the north, there's massive halimeda mounds, 20 metres, 25 metres thick, between the coral reefs. And the information you showed here is, well, they're aragonitic, we knew that, and they don't seem to have much ability to withstand ocean acidification. Is that correct? Do we expect to see them decline?
Look, there's been a fair bit of work on halimedas exposed to ocean acidification. Overall, my impression is that they're much more robust than what we initially thought, and that's potentially because they are aragonitic. And aragonite is much more robust than the high magnesium calcite. So I don't think halimeda would sort of decline dramatically due to ocean acidification, but I guess that would depend on the control of the living alga on those sort of banks that have already been sort of where there is no living algae. So those skeletons on those bioherms in the northern Great Barrier Reef, which are on the top covered by Halimira, but on the bottom they're just dead Halimira. Those meters, which are sometimes hundreds of meters of Halimira banks, could be more prone to dissolution, but the upper living layers of those Halimira banks could potentially be still sort of doing well under OA conditions. So, yeah, that's as much as I can tell you here.
All right. There's one more question before we break.
Thanks. Thanks for the talk. It was very interesting and enlightening. And it's just a comment that, you know, when we measure the carbon chemistry, we always calculate the saturation state of calcite and aragonite, and never see, well, most of the times we don't think about dolomite, which is more soluble. So those who calcify dolomite would be like more sensitive, maybe. And I think it's good to think about that, and from now on also include dolomite in our calculations and in our projections as well.
Yeah, definitely. That's one of the gaps that I see in the research on coralline algae, because most people really use the saturation state of aragonite for their studies, but we don't see a lot of saturation state of high magnesium calcite with respect to seawater, which I think would be a much more relevant metric to understand whether the algae are on those thresholds of whether they should thermodynamically be precipitating or dissolving the skeleton. In our lab, we use omega high magnesium calcite for the coralline algae. But in terms of dolomite, you could also calculate that, the omega for dolomite, and dolomite is a much more robust mineral, probably many, many times more robust than aragonite. It's a mineral that probably very, very hard to dissolve with projected CO2 conditions. Mm-hmm.
All right, I think we're...
Coffee time.
So that was a really fantastic talk, and just wanted to thank Guillermo again. Okay, it's break time.
By the water neath the Mexican sky, drink some margaritas by a string of blue lights, listen to the mariachi play at midnight. Are you with me? Are you with me? I wanna dance by the water neath the Mexican sky, drink some margaritas by a string of blue lights.